So our goal, really, if you look at the top of the screen, was to focus on what we call functional genetic variants. So these are more likely to be genetic variants encoded in what's called exons. So these make up our proteins. The introns tend to include kind of more regulatory function, but our hypothesis was exonic variants were more likely to be involved with Chiari. So we had two major questions. First of all, when we compared individuals with and without Chiari, do we see differences in the number of variants in these genes? And then our second question was to see if we look within the Chiari patients, those with connective tissue disorders and those without, do we see a difference there? Next slide. So let me tell you a little about our data set. We included not quite 200 non-Hispanic white adult women. Next point. And we assessed essentially whether or not they had connective tissue disorder symptoms, either from the Biden scale or that there was documentation of symptoms that were consistent with CTD. Next point. So what you will see here are the patients that were sequenced were comprised approximately 50-50 from the Chiari 1000 project and Duke. Next couple of points will show you that there were some differences with the CTD positive percentage across the two groups. There really wasn't a difference related to those with or without a formal EDS, Ehlers-Danlos diagnosis, and there were some differences with respect to Syringomyelia, but it wasn't very drastic. Okay, next slide. So overall, after quality control and processing, we ended up with over 1,300 variants in these 21 genes, but a very small proportion of those, nearly 500, were actually functional. So those were the ones we wanted to focus on. Next point. But when we started looking at these variants and comparing them to the literature and the public data, what we found was there were a lot of those variants that really were present in all these other databases. So they really just were not likely to be associated with Chiari. So we excluded those. Next point. So this graph shows you just for every gene how many variants we identified. So what's probably immediately obvious to you is Some genes had a lot of variants and some genes did not. So I point this out just to say that a lot of this simply has to do with the size of the gene. But what we were really interested in were the variants encompassed by those red bars. Next slide. All right, so now getting to our actual research question. So the first question was, do we see more rare functional variants in these genes among Chiari patients versus controls? And so while we only had about 200 Chiari patients, we had over 60,000 controls. So there was a lot of control information. Next point. So the most exciting news is we actually did see several genes for which there was an excess number of these potentially functional rare variants in Chiari patients compared with controls. And so, you know, I'm sure all of you don't know what these gene names are, but really the first four of those are collagen genes. So these help make up the extracellular matrix, and variants in these genes are often associated with connective tissue disorders. The last two genes are part of what we call the VEGF pathway, and those genes are involved in vascular differentiation. So we'll talk about a little later why those might be important. Next point. But what was really surprising to all of us was for some of the genes, there was a large percentage of our patients who were harboring a variant in those genes. So over a quarter of the patients had a variant in that collagen 6A5 gene. So this was really quite a surprise to us. Next slide. So our second question then was, if we restrict this analysis just to Chiari patients, how do patients with connective tissue disorders compare with those who do not have connective tissue disorders? Next point. And so what we found, next 2 points, was that for the CT positive subset of patients, they actually had a lower percentage of patients in that collagen 3 gene, but they had a higher percentage of variants in the collagen 7 genes. And what is also perhaps important to say, although we didn't have a large percentage of documented Ehlers-Danlos diagnoses in these patients, we did exclude those patients, reanalyzed this data, and those associations held. Next slide. Okay, so again, you know, why are we excited about some of these genes that have been implicated? So I already talked a little bit about the collagen genes. So these are really exciting because they have been implicated in a number of conditions that often co-occur with Chiari malformation. And we know they're important for bone formation and growth and connective tissue. Next point. So then the second pathway that was implicated was that VEGF pathway I mentioned. So why might vascular genes be important for Chiari? Well, because we know that vascularization really is very important for bone formation. So that's what we hypothesize those genes may be doing. Next slide. So let me just tell you, I'll wrap up kind of quickly here, what some of our future directions are. So we're really interested in trying to figure out functionally what these genes and what these variants might do. And some of the ways we can do that is maybe to create some animal models and see how the bones develop in those animals. We're also, within the human data set, very interested in looking more closely at craniomorphometric traits. So a lot of the patients that were in this study had craniomorphometric analysis. So we're really interested in kind of overlaying that information. And then finally, for the future directions, we know we only looked at a small portion of the genome. So we would like to expand that to look at many other genes. Next point. Right, so basically, at the end of the day, what our study told us is, just as we had hypothesized in the beginning, there are multiple genes which are involved. And there's even different pathways, but a lot of them seem to have reasonable connections. Next point. And just as some of our previous work has shown, we do still believe that the presence or absence of these CTD symptoms is directly associated with the underlying genetic mechanisms and ultimately the biologic mechanisms. Next point. And so again, just to remind everyone, we feel like the collagen genes are going to be really important, as well as that VEGF pathway. So hopefully that gives all of us, not only our group, but other researchers, something to kind of start exploring and trying to understand a little bit better. So the last slide is just my acknowledgements. And so again, thank you all for